Okay, I'm back. I started recording also. Let us discuss about translation. This I already told. So translation based means nothing but, two amino acids. The overall structure of amino acids. From the name, you can understand that amino and acid, like amino group and acidic group, both are present. This group and this is the COOH, right? The acidic group, and one H atom that is also present in every amino acid. So we have 20 types of amino acids naturally available in our body, and one is R group, the side chain. So this we have 20 different types of R group, and that's why we have 20 different amino acids. The simplest one is H, and then it will be CH3 and much more complex. So if this R H, that is called glycine. Now, two amino acids, they are like together through peptide bond. This is peptide bond, and one water molecule will be released from this system. This is called condensation reaction. Remember, in order to carry out this reaction in chemical lab, it's required lot of lot of effort. I would say you might need or high temperature and so many things. But think about this reaction is happening in our body naturally, right? In ambient temperature, our body temperature, we have like lot of alcohol, acid, not organic solvent, all those things, but still it is going on, right? Because of enzymatic action, because of physiologically relevant system. So ribosome helps in translation, right? So let's talk about it. Here, those are the just schematics that 1, 2, 3, 4, those are the amino acid number. They are attached by peptide bond. So as a result of that, for example, if I say tripeptide, tripeptide. So don't confuse that. In tripeptide, we don't have three peptide bond. In tripeptide, you have three amino acid and two peptide bond. Little bit confusing, that's why I told tripeptide, dipeptide, two amino acid, one peptide bond. Don't forget. Now, the process of translation. This one I would like to help of whiteboard first. First, I will make the concept clear. Then you will understand whatever I am trying to say in that slide. So, translation, I already explained the term. So here I'm writing something, right? So if I write something like this, let me think. So I can see my whiteboard. So something is written here, but the problem is if you don't know this language, whatever I'm writing, like, for example, in this case is English letter, so you can at least read it. But if you are not aware about it, how you will decode the information? How you that's the main problem. Because in our gene, we have just like some like sequences, just eight order of sequence, the permutation, combination, all those things that makes the case complex. And then you are getting the mRNA, and from this mRNA you have to make the protein. Before that, you need to you need to have some conversion mechanism, that some language, like both the language can be, understandable by our system. Similarly, in this case, I'm writing something here, but it's hard for you to understand what I have written here for two different reasons. The one thing is, if the language to you, and the other thing is the way I have written this. That's the problem. So since all of you are aware about this language, so I can go a little further, and then I will correlate this with our translation system itself. So because you studied at the very beginning at your school the English, in this case you can just divide it like this. Intentionally, I didn't put any gap in between. Okay, so now you have some meaningful sentence to understand something here. Before that, if you cannot understand what is written here, in order to get the protein, you have to understand the mRNA sequence and then convert that. So the sun had set but not out yet. So if the whole thing, then you are getting the sense, right? What is going to happen, or what's the meaning of this sentence, right? That is now done. Now, only thing here intentionally, I have selected as I would say all the words are three-letter word, right? All are three-letter words. Similarly, in our genetic system, while we are making protein out of mRNA, so if it is an mRNA, 5 prime, A, U, G, C, G, C, G, C, G, something like that. Just randomly I wrote. So now you don't have T, instead of that, forever. So the mRNA sequence is going on. Like you have multiple sequence here. I'm not writing, okay, in between here. So you have a lot of sequence. So this letter, which was giving you some meaningful word like the sun, set, not. So you have some sense here. So similarly, in genetic system, in our gene, in our mRNA, those three-letter code, they are also some meaningful, they are carrying some particular meaning. So these three-letter nucleotide together, this is called codon. Remember that this is called codon. So this is codon 1, codon 2, 3, that way it's going on, right? This is called codon. Now the ribosome make protein. So if you have one mRNA here, big and long mRNA, 5 prime, 3 prime. So ribosome is here, this is ribosome which make proteins, and it will move in this direction. You have many sequences here, right? Like U, C, A, C, A, U, G. So ribosome is moving there. So while ribosome is scanning through this mRNA, whenever it encounters first AUG, the first this letter AUG, that is called the first codon. So from that point, protein synthesis will be started. And that defines this. Because here we are just writing some sequence like UCA, any sequence. But whenever it will select the first codon here, AUG, the start codon, then every consecutive 3 nucleotides present on mRNA, that will be the next codon. So that way it will go on. Now, I should tell you that in every living form, whether it is bacteria and animal, AUG is the start codon. From the evolution perspective, people say, like scientists believe that is again suggest translation is a fundamental mechanism. Every life form, they need to make protein, right? So as a result, this is very fundamental mechanism. And if you see, in every organism, whether it's plant, animal, or bacteria, AUG, that is, we evolved maybe from a common stock. That's kind of an idea. So now I have few questions for you that can be without biology. So tell me quickly, how many codons should be there in our body? Codon means how many different types of codons should be there? It is a simple question, right? If you know what is codon, which I already explained, how many codons should be there?
64.
60. Because 3 nucleotides that makes a codon. And we have 4 different types of nucleotides. 4 into 4 into 4. So we have 64 different types of codons, and out of that, which is always start codon, and 3 stop codons there. So one is UAG, another one is UA, and another one I forgot. So also you don't need to remember this thing. I'm just explaining. Okay, if I like ask something like this during your information. So AUG is the start codon, and we have 3 different codons which are called stop codon. We have 64 codons. Out of that, one start codon that always bring methionine, that is an amino acid. Out of 20 amino acid, one is methionine. So now you can understand that means all proteins in our body, they start with methionine, right? The same amino acid. And 3 stop codons. So that means stop codon, they don't bring any amino acid, no amino acid. So that's why translation stop. The translation, that means the protein will not be synthesized anymore. It will be done here. So that means 61 codons that include start codon that brings different types of amino acids, right? And 3 codons, those are stop codons. Now, I that we have 20 different amino acids, like 20 different amino acids. That means again one degeneracy present here. Degeneracy. You can tell that multiple codons can encode or can bring just one amino acids. That's why we have one codons, they code for, they specify 20 different amino acids, right? So that's the idea. Now, let me go through it peacefully. I hope you will get the point. Now, then again, I will explain wherever the problem will be happening. So no, I explain, then I will go into it here. Now the point is how this conversion is happening. That from nucleotide sequence, that means from codon to amino acid sequence, that I should explain. So in this case, for translation, you need to know about 4 different important things. One is the template, that is mRNA. mRNA for a long time, this is mRNA. Then the machine that translate, that is ribosome. Ribosome is a big complex. This is ribosome, which is made up of different proteins and ribosomal RNA. all together they form this complex that is called ribosome. And one more thing, tRNA, that is called transfer RNA. Transfer RNA. So tRNA, and inside ribosome you have rRNA. Last week also you saw that mRNA, messenger RNA, that had the message to bring the protein. rRNA, ribosomal RNA, that constitute the ribosome. tRNA, transfer RNA, that transferred the message. So tRNA that helps to bring particular amino acid. How? That I'm trying to explain here. If this is the sequence here, 5' AUG CGU and something like that. Now, this is the mRNA sequence. The tRNA or transfer RNA, that is also RNA molecule, but which is little bit shorter. It looks like, say for example, something like that. Now, I told you that this is codon, like 3 nucleotide, this is codon, and this is codon, codon here. And in tRNA, tRNA is RNA, so as a result of that, this is also polymer of ribonucleotides. This is codon, and the sequence present in this loop is called anticodon. So as a result of that, in this tRNA, you have what sequence should be there? Again, similar Watson-Crick base pairing. So in this tRNA, you have UAC. So that's why they form Watson-Crick base pairing. But this particular tRNA always bring methionine. That's why start codon always bring methionine. So this will be selected by this codon. And ribosome is present here where the reaction is going on. Ribosome helps to catalyze the peptide bond formation. Peptide bond between what? Peptide bond is being formed between 2 amino acids, right? So as a result of that, the next amino acid, that means here you have an tRNA. So this tRNA will bring another amino acid. Okay, another amino acid. And here in the anticodon loop, this is the anticodon loop. C, so here it should be G, C, A. So this is the anticodon sequence present in this tRNA. And tRNA will bring a specific amino acid. I don't know which amino acid should be coming here because you don't need to remember, right? So, but one amino acid will come. Two amino acid will be very close to each other, and now ribosome will make the peptide bond. Ribosome is moving in this direction, then the next one will be the correct amino acid, and then it will be again attached to peptide bond. So this way it is going. So basically, you are transforming the information which is present on this mRNA. You are making a protein sequence, and this protein finally it will fold into 3-dimensional structure, and that will give you a particular function. So that's the idea. Okay, so that way protein synthesis takes place. So major like things you need to understand a little bit is mRNA that contains the message. That means those codons are important to bring particular amino acid. Now who will bring the amino acid? tRNA, transfer RNA. So tRNA, they are also called adapter, because if you in tRNA, like in this case, see here, through one portion, it recognizes the particular codon. Another side, it is bringing an amino acid. So it is actually, I would say, it's act as adapter molecule. long time back, actually Crick, before people were not, scientists, they were not aware about this step, how this conversion happening. And Watson and particularly Francis Crick, he proposed this. This is called adapter hypothesis. So he suggested that there should be some molecule which acting as an adapter. At one side, it will hold the sequence of mRNA, that is codon, and through other side, it will bring amino acid. So that is what is happening here. Because if you see adapter, all of you know, like when you cannot charge your computer because of a different type of plug point. Right. Similarly, in this case also, while you are converting this information from nucleotide sequence to protein sequence, this tRNA, they act as and that help the conversion of this information. So that is all about translation, the basic about translation. Let's see what is present in this slide. As I told you, the protein synthesizing machinery, you have ribosome, ribosomal RNA and proteins, and it is complex structure. You don't need to remember all those information mentioned here. These ribosome structures discovered by many scientists, and 3 of them they obtained Nobel Prize in chemistry in 2009, as I mentioned a few minutes back. And Venki Ramakrishnan was, and another scientist from Israel. Her name is Ada Yonath. So she came here at IIT Kharagpur in 2017. Ada Yonath from Israel. So she also got the Nobel Prize along with Venki Ramakrishnan. Okay, like contribution to understand the ribosome structure. So anyway, if you see now, this is that is messenger RNA. And now, if I extend the sequence of this messenger RNA, this is the 5' end and prime and sequence are mentioned here. Now, as I told you, when ribosome will scan, the first 3 letter, which will be like, which AUG, that is the start codon in this case, and every consecutive 3 letters just after that, those are the next codon, and another codon, that way it will be going on. And the first codon brings methionine, and the next one AAG, that codes for lysine, arginine, proline, that way it's going on, and you will get, that means multiple peptide bonds present here, right? So proteins are of varying length. It depends on from which gene it is being synthesized, how is the gene, how is the mRNA. So proteins can be like just 100 amino acid long. It can be 5,000 amino acid long. That is possible. It can be 300 amino acid long. It can be 100 amino acid long. As I told you, we have more than 1 lakh different types of protein. So you can understand they are of different types. And also, if you see, 6 billion, the folding overall structure of DNA is same. Like that's why if you will see that people, they are saying that, okay, I'm working on DNA, structure of DNA, not that many labs work on it. But protein, yes, because all these different types of protein, they are polypeptide, but polypeptide in linear form, they are not functional. They need to fold, need to be folded properly into 3-dimensional structure, so that they should attain a particular function. So in their 3-dimensional form, they can perform their specific function. Okay, this is called protein folding. So from the next class, Dr. Somo De, he will be taking classes and he will explain this protein, how this linear sequences of amino acid converted into 3-dimensional structure and how it helps to attain a specific function. But the thing what we were discussing here, that you are getting polypeptide. So now the question is how the ribosome will recognize template or mRNA. In case of bacterial system, in case of E. coli, prokaryotic system, some sequences are present, which is called ribosome binding site. So ribosome will bind to this site and then it will move in this direction. And the first AUG will be taken as start codon. But in prokaryotic system, instead of RBS, or ribosome binding site, 7mg cap, which I discussed, at the beginning of this class. So that is recognized by the ribosome, eukaryotic ribosome, and then it can start the translation. And if the ribosome moves, this peptide, and it should be detached from the ribosome when the stop codon appear, and this nascent broad polypeptide will be folded to give you a protein, and it will be functional then. And tRNA, I already mentioned this anticodon loop that helps to recognize a tRNA. They bring a specific amino acid. So as a result of that, you can, or we are able to convert this information from sequence to amino acid sequence. Now, if I ask you like very general, I would say like general question. So what is the nature of interaction between codon and anticodon, or tRNA and mRNA? Basically, like properties of interaction means I'm trying to ask you what kind of interaction involved there. You can tell, like between codon and anticodon, what kind of interactions happening? I already, like, in the whiteboard, I was there. So at that point, is it possible for you to say what kind of interaction means? I'm asking what kind of bond, whether it's covalent bond, ionic bond, something like that. You don't need to remember anything to answer this question, right? So what kind of interaction between codon and anticodon sequences? Tell me. Is this covalent bond or non-covalent bond? First, tell me that.
Sir, hydrogen bond.
Sir, hydrogen bond.
Yeah, very good. Right, because it should be like, dynamic, right? It's a covalent bond, it will attach and it will stay there. No, the ribosome is moving, and it is just bringing tRNA. So this bond is just to bring the correct tRNA with the amino acid. Because if you see inside the cell, like when we are discussing, we are just showing one mRNA, one tRNA, just 1:1 kind of interaction. But inside the cell, you have to appreciate that lot of like huge amount of like macromolecular concentration. That means a lot of molecules nearby. They are in Brownian motion. They are hitting each other. But still, the correct amino acid at in the nascent polypeptide. And how it will be selected? That is being done by this codon and anticodon recognition. And this is just base pairing, just hydrogen bonding. So it's bringing the correct tRNA. tRNA is related to a specific amino acid. So as a result of that specific amino acid sequence, you will. The problem in this case, like some wrong amino acid, it can destroy the whole protein. Some mutation in some gene that can destroy the whole protein. Protein sequence, you cannot put some amino acid. It will completely destroy the protein. It is very sensitive, I would say. So now, if you see here, so here some chemical reaction mentioned here. Just you don't need to go through it, the all details. But what I want to tell you that a specific type of enzymes are present that help to attach particular amino acid to a particular tRNA. So those are called tRNA when they are attached to amino acid. And here you can see the codon and anticodon base pairing, whatever I just discussed. If you have U, the codon then should be AAA in the tRNA, and that will bring this amino acid. This is phenylalanine, another amino acid out of 20 amino acid. This is one of them. And those are the some important statement which I already discussed, right? So here in Quran, I already told you also, and whatever the concept of genetic codon, how many codons present, all those things I already mentioned. And this is the genetic code chart. So this is not for you. This is people who are working with this kind of problem in the lab. Time need to change the codon to make a different protein, some mutation. That time we follow this chart, just like periodic table here, codon chart, that which codon is bringing what amino acid, and based on that you can design your experiment. I told you that AUG is the start codon that brings methionine, and AUAG and this one, those are stop codon. This stop codon sequence you don't need to remember. And this is in just one slide. Lesson and involved machineries and processes. Whatever I tried to explain, you will get overall idea here. Very briefly, I'm discussing. So the mRNA will be recognized by part of ribosome that is called 30S ribosome, and then the next tRNA will come. tRNA, this is the anticodon, and it's bringing the first amino acid, that is methionine, and then in the next step, the other portion of the ribosome join. This is the complete ribosome, and now it will bring the next amino acid, and as a result of that, those two amino acids should be close to each other, and ribosome will catalyze the reaction. So as a result of transformation will take place, and now this ribosome will move further in this direction, like in this direction. So as a result of that, you can, this is the previous position of ribosome, and now it has moved here. So as a result of that, again, another tRNA will bring amino acid. So another will find here. So by that way, it will add one after one amino acids, right? And the protein will be synthesized. And this is the step involved in the system, like replication, then transcription, and then translation, everything summarized in one slide, right? Now, I have one small question. I told you that in bacteria, transcription and translation, those two are coupled process. That means they are happening at the same time, but not in eukaryotic system. So as a result of that, I ask that if the transcription rate in E. coli is 45 nucleotide per second, the transcription rate in E. coli is 45 nucleotides, then what will be the translation rate? What do you think? Just, whatever you understood, on the basis of that, what could be the answer? Will be the translation rate in the bacteria. Any one of you, just try to answer. Whatever you learn, based on that, you can analyze this actually. Like I'm telling you that if the transcription rate is 45 nucleotides, then what could be the translation rate? So whether that can be mentioned, or whatever you have learned, it can be anything. What do you think? So since transcription and translation in bacterial system. So as a result of that, if this is the case, this is mRNA 5 prime, 3 prime, this is going on, and now translation going on by ribosome. This is translation because happening together in E. coli, I told you. So if the transcription rate is 45 nucleotide per second, and also the translation rate is 45, then it will hit here soon, because 3 nucleotide code for 1 amino acid, right? One amino acid, and 1 codon means 3 nucleotide. So the process is, I would say, 3 times slower. So the result of that, if it is 45 nucleotide per second in bacterial system, approximately the translation rate should be 15 per second. Okay, so that it can keep, I would say, it is continuously going on. The transcription is going on here, and translation is. This ribosome will not hit in this system, and it will not, make a problem here. So as a result of that, the translation rate is almost 3 times slow compared to transcription rate. I hope you understand. Now, the next question is, like interesting question. Which came first, nucleic acids or? So those are the basic concept of flow of information. That means of molecular biology, like replication, transcription, translation. But you are getting protein at the last step, right? You are getting protein here only. Translation. But remember, this DNA polymerase is a protein. RNA polymerase is also a protein. Then how those formed initially in terms of evolution, if you say that which came first? This is a difficult question to answer. This is all like this problem. All of you are aware that which one is first, like chicken or egg, something like that. So at this point, although you are like participating in discussion, but I'm just like to ask you one more time, if you want to suggest something like which one is first, do you think, like or nucleic acid? What could be your answer? Like anything, like it's nothing, I would say right or wrong. I would just want to see how the information. So which could be like, which came first, nucleic acid or protein? What is your answer?
So protein, at least the Miller-Urey experiment suggests that.
So then if protein is first there, so then that protein should be coming from some right? So as a result of that, you need to have mRNA also, right?
So probably
So,
they were not from translation, they came through separate chemical reactions.
yeah, okay. Okay, it's good that you tried. Definitely, if I'm in your place, I would also say the same thing, that it's protein. But what about if I, if I say some nucleic acid, they might have enzymatic activity, right? Say, like they have the enzymatic activity, like they can work to some extent, although most of the time I told you guys that DNA, they are strictly informational, RNA, they are operational, and protein, they are actual functional molecule. But some nucleic acid, but RNA, they have some enzymatic activity also. Okay, so as a result of that, like one big theory there, not big or like big textbook also there, that is called RNA world hypothesis. Neither DNA nor protein, they are the beginning molecule in a live form when it's kind of cell and organism came. So it could be RNA, which was the initial macromolecules before DNA. RNA can act as genetic material. Additionally, RNA can act as an enzyme also. And if you see, both things are present now also, that reminiscent. For example, some of the viruses, they have RNA as their genetic material, like for example, influenza virus, for example, the SARS-CoV, whatever happening now, right? Those viruses, they have RNA as their genetic. But the problem of virus is they are considered as non-living because they don't have protein synthesis machinery. So they first need to infect host, first need to go inside the host, and then they utilize the host machinery. They don't have ribosome, they don't have tRNA. They use our protein synthesis machinery. For that reason, virus, they are present outside of their host, they are considered as non-living particle. Whenever they are coming inside the host, they are acting or are multiplying, they are utilizing our hosts, like various component of host. So that means RNA can act as genetic material, although like most have DNA as genetic material. Similarly, RNA can act like it can perform some enzymatic action also. For example, the splicing I told, to remove the RNA, like intron. There also RNA itself has the enzymatic activity. And the most fundamental mechanism is protein synthesis. You will try to know that the actual catalysis, the peptide bond formation, right? The actual catalysis is being performed by ribosomal RNA, not by any protein enzyme. It is RNA. So again, it is the proof that RNA could be the beginning. It has enzymatic activity also. Everywhere in any life form, the translation, the most fundamental event, that is catalyzed by not proteins, though all other enzymes are proteins. But in this case, ribosomal RNA itself catalyzed the reaction. So that again proves that RNA could initial molecule. And see here what happens inside the ribosome. You don't need to go through the details, all of those things, but what I want to mention, in ribosome itself, I told you have multiple proteins and ribosomal RNA. In three-dimensional structure, ribosomal RNA, a few nucleotides are close to each other and they form the active site. So, discovery of ribosome structure by Venki Ramakrishnan, Ada Yonath, and Tom Syke, they actually, and that's why they have been awarded Nobel Prize in 2009. So this is the active site, and it helps the peptide bond formation and transferase reaction, and that help in peptide bond formation, and that is happening everywhere in bacteria, eukaryote, plant, everywhere. And this is correlation between transcription and translation. This slide, again and again, I told you that transcription and translation, they are happening together. And in case of eukaryote, those two are separate process. And here, the translation machineries are attractive target for therapeutics. This is the last slide today. So there are many antibiotics which target translation machinery, many antibiotics like tetracycline, chloramphenicol, which are predominantly used in eye drop. So they also block protein. And similarly, streptomycin, erythromycin, those are actually generic name, but if you go to medicine shop, they will give you many antibiotics. Might not be the same name, the ingredient should be something like that. But here I have something to address and then I will be done today. The point is antibiotic. So those antibiotic, whatever I just mentioned in that slide, for example, chloramphenicol, which is like present in many antibiotic eye drop. So that the bacterial ribosome, not our own ribosome. So that while scientists, they are designing antibiotic, you have to carefully design something which will prevent the action of some bacterial portion. Like if you particularly block the protein synthesis, that means bacteria cannot survive if you stop their protein synthesis. So as a result of that, when someone is sick, you are taking antibiotic. So that antibiotic should not have your own synthesis. So as a result of that, there should be some differences between bacterial ribosome and our own ribosome, right? And on the we have designed antibiotics so that it will specifically block bacterial protein synthesis, not our own protein synthesis. So that how these antibiotics work. So there are many different types of antibiotic. All the time they don't block protein synthesis. Sometimes they block cell wall synthesis also. But antibiotics, either they block protein synthesis or they block cell wall synthesis. Now, the another important thing you should uh, you should not take antibiotic randomly. That means you are not at all sick, you have no problem, you shouldn't take a lot of antibiotic. That's very bad idea because in our body we have a lot of good bacteria, right? Many. You cannot like imagine how much good bacteria we have in our heart, mouth, everywhere. Lot of good bacteria. So now when you are taking antibiotic to kill some bacteria you are might be little bit of sick, but when you are taking antibiotic, that is like killing all sort of bacteria present in our body. So that is clear those bacteria also. That is not a good idea because those bacteria, they are actually helping us. They are present in our gut, in our like various portion of our digestive system, so that pathogenic bacteria which can cause disease, they cannot colonize, they cannot sit and infect quickly. So those bacteria are helping us. But if someone is taking antibiotic without reason, that is creating problem to those good bacteria. Okay, that's why it is advised if you have requirement, then only someone should take antibiotics, right? That is the idea. And as I told you, just 1 minute I will take that you will be to know that how many bacteria present in a healthy individual. If someone is really healthy, then if I have like 100 cells in my actually we have 10 to the power 13 cells approximately in adult. If I have 100 cells, 90% of that is bacteria. So that is small, but absolute number of those bacteria, that their number is much more than our own cell number. And those are helping. They are important. They are helping us, but unnecessary when someone is taking a lot of broad spectrum high dose antibiotic, actually also killing this good bacteria, and that is kind of sometimes destabilizing our health, and it will take some time, and again those bacteria will be propagated and then will right. So that's all. So by that way I'm done. So from next class Dr. Somo De, so he is all from our school of bioscience, so he will be taking your classes, and I'm done. Thank you very much, and you can leave.